A very warm salutations to all. Myself Param Malik, a student of Kerodimal College, currently studying BSc Chemistry Honours, and I am here to make you even better understand the topic of amino acids and its classifications. So, before we start the topic, we have to get through the actual roadmap of what to study in order to master this topic. First, you should have a basic understanding of the amino groups, the carboxylic groups, the alkyl groups. You should be able to identify and classify by side chain history. You should have the basic knowledge of polarity, non polarity, acidic, basic, aromatic, and special sulfur containing bonds. You should be able to interpret pKa and ionization physiological charge, and also some disulfide stabilizations and all the other points mentioned in this topic in order to have a mastery on this topic. So without any further ado, we should study and get to the first topic. The first topic we have is fundamental structure and nomenclature of amino acids. So before starting the topic, we should know what are amino acids. Amino, basically the NH2 group, and acids, basically the carboxylic group. So what are amino acids? A compound having amino group and a carboxylic group bound together by a side chain. So this side chain determines the qualities or the traits of the group. Like glycine, it has a different number of CH present in the side chain than from alanine, so they behave differently. When we look at the structural diagram, we have a simple formula NH2CHRCOOH. As I told earlier, one amino group and one carboxylic group bounded together by the side chain. Zwitter ion at physiological pH. Basically, Zwitter ion is nothing but a compound having positive and negative charges at the end is the definition of Zwitter ion. So nomenclature or the scientific naming, when it comes to it, we basically prioritize the acid group rather than the amino group. So we start the numbering from the acidic group. As you can see in this one, in the structural diagram, as we have given NH2CHRCOH, when we go to name it in the nomenclature on the scientific manner, it becomes 2-amino carboxylic acid. So why 2-amino? Because first carbon is the COOH1 and the second CHR. To that is bonded the amino acid. So 2-amino carboxylic acid, because we prioritize it, carboxylic acids. Okay, what are the keynotes? There is a special feature. Out of the 20 protogenic amino acids, all the 19 are chiral carbon, which means they are optically active. Only one is optically inactive. Can you guess which is it? It is glycine. So going on to the next part, we have peptide bond formation, condensation, and primary protein structure. So what is peptide bond? We have one amino acid, one monomer. Let's say glycine, alanine for example, take any one, valine. They are a monomer. They have to be bonded together through the N terminus or C terminus or the NC bondage or the peptide bondage. So what is bondage of N terminus and C terminus? N terminus is basically the end of the NH2 group. Like in the compound, when we start from the amino side, it is the N terminus, and the ending molecule or the ending atom, C terminus, is basically the carboxylic group. So we start from N terminus and end at C terminus. When these monomers line up together, they are bonded by C and N terminus bonding, which is primarily referred to as the peptide bondage or the amino acid linkage. Carboxyl of one reacts with amine of other, releasing H2O. As you can see, it is written, and by the diagram you can clearly show the peptide bond in which resonance stabilized amide linkage is formed. So coming to the next part, stereochemistry of amino acids depending on L and D isomers. So what are L and D isomers? L and D are short forms for levo and dextrorotatory isomers. Levorotatory basically rotate the polymer to one side, whereas the D or rotate it to the other side, which makes the compound chiral or optically active. So chirality at alpha carbon. Most protogenic amino acids are chiral. Glycine is achiral because it does not have a chiral carbon. L versus D configurations. Proteins use L amino acids. Well, Fisher projection shows NH3 plus on left. It's a good way to represent L versus D configuration. We can also use wedge-3D models to get a better grasp on the visual models. Again, there is one exception, glycine, because it is achiral. Coming to the next part, we have amino acids and classifications. So depending on the variety of side chains that bound the amino group and the carboxylic group, we have 20 majorly used amino acids. We have the main names of the amino acids, then the representation can be done by two methods. First, there is a 3-letter representation and there is a 1-letter representation. Like for alanine, 3 letter is ALA, 1 letter is A. As you can see in the list, there are many. Just go through the list as I pause, and don't get mixed up because some are similar, like glutamine and glutamate, argentine and argentate. So better not confused between the one word and three words of the amino acids. Now, what are the types or classifications of amino acids? So basically, the first type we have is the non-polar one. Non-polar amino acids. So amino acids which have residues, properties and typical locations. Okay, key examples. Residues are basically the amino acids that are left. Example, glycine, alanine, valine, leucine, methionine, propene, tryptophan. Properties, as you can see, they are non-polar or hydrophobic, which is generally describing itself. Non-polar means they lack the strong hydrogen bonding. They prefer interior of globulous proteins. They don't have powerful hydrogen bonding, so they bend and fold and thus form hydrophobic effect, which is nothing but the folding of the main chain. Functional notes: aromatic residues enable stacking interactions. Aromatic residues like tryptophan enable stacking interactions because they have ring, and also TRP or tryptophan is a strong UV absorber and in spectroscopy. It is a functional note. So yeah, these are some of the exceptional qualities can be seen in the non-polar ones. When we go to the polar uncharged amino acids, which is self-explanatory. Polar means these are charged. They form hydrogen bonds. So residues are nothing but the amino acids which fails to react, but most of them are covered under polar uncharged. Residues: serine, threonine, glutamine, tyrosine, commonly polar. Properties: as you can see, they form strong hydrogen bonds. As they can form strong hydrogen bonds, they are more soluble in the solvent is basically used water or maybe any other. Frequent on surface and inactive site networks, as they form hydrogen bondings inter and intramolecular, so they have inactive site networks. So what are some of the main functional notes we have to keep in mind? In serine or threonine, OH act as a nucleophile. It can also be explained through the serine proteases. Also, in ASN or GLN, stabilized structure is formed via hydrogen bonding. Tyrosine is both polar and aromatic. Cysteine can sometimes also be polar. Coming to the next part, acidic and basic classification. First, let's start from the first. First, we saw non-polar amino acids, then the polar ones, and now the third category is of acidic and basic amino acids. So residues, as we can see in the list, we have aspartic acid, glutamic acid. We have their pKa values in which they are most stable, or their pi value, which is also isoelectric values. So physiological form at the pKa value of 3.9, aspartic acid is deprotonated, negatively charged. Basically, due to the charge on the COO minus, as we comes down the list, we can see Rg9, which is 12.5, which is highly basic, so protonated, and positively charged due to the charges on the amino group. Similar fashions can be listed in the list, so carefully note them. And as we go forward, we have some special case amino acids or functional effects. So let us study them. First type, we have the aromatic type. Under this, certain examples of amino of 20 majorly amino acids that we represent are phenylalanine, tyrosine, tryptophan, absorb UV. They can absorb UV, right? But TRP is strongest, which means TRP absorbs the UV strongest at 280 nanometers. It gives it lambda max at 280 nanometers. The second type is the sulfur containing. Cysteine thiol forms disulfide bonds. So cysteine in its thiol forms form disulfide bonds which also stabilize the structure, which is tertiary in nature, and also methionine in its thioether form, it serves as start codon in protein synthesis. It plays a major role in the start codon. Okay, so going on the third part, we have proline, secondary amine that imposes conformational constraint. It acts as a basic function, so it also introduces kinks in helices. Helices is nothing but the plural of helix. There are two kinds of helix: single stranded and double stranded. We will observe them in the next classes. So as we go forward, we have a simple classification of essential versus non-essential amino acids, dietary needs and biosynthesis. First of all, we need to see what is the difference between essential or non-essential amino acids. First one, essential, which cannot be synthesized by the human body. For example, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, tryptophan, etc. Adults require all 9 in diet. These all 9 amino acids are required by adults to be absorbed because they cannot be synthesized in the body. And as far as we go for the non-essential amino acids, we call them non-essential because they can be produced in the body and does not to be required to be they are only synthesized in the human body and does not require any external frame to be absorbed through via, which is basically food. So they contain alanine, asparagine, aspartate, glutamate, serine, glutamine, proline, glycine, tyrosine. Tyrosine is conditional. It can also be obtained through food or synthesized in the human body. Tyrosine synthesized from phenylalanine. Some became conditional during stress, so tyrosine also helps in stress management. Classification summary table, as we have studied earlier, we have met or seen or studied several types of classifications. First one we go through the non-polar one, which are hydrophobic in nature. Then we studied about the polar amino acids, which have strong hydrogen bonding. After that, we studied about the acidic and basic, their pKa values, and which have positive charge and negative charges depending on COH group and NH2 group, and the pKa values they are more stable in. Then we studied about some special classifications like aromatic group. They absorb UV, they have stacking interactions. We also saw some sulfur containing bonds like disulfide bonds are formed in cysteine, and we saw the essential and non-essential amino acids, like how a human need to consume both, but which are produced in the body and which are needed to be absorbed via external source, that is food. So as we go forward, structure to function case studies, the amino acids that we have studied till now do not act independently, rather they act as a group and also help each other. The best examples can be said about the serine protease catalytic triad. It has 3 amino acids, basically serine, histidine and aspartate. Its best example is chromotripsine. Serine acts as nucleophile, histidine as base, and aspartate stabilizes histidine. So they all 3 help each other to have a stable ecosystem, also known as chymotrypsine. Okay, the second case study we have is of insulin disulfide stabilization. In its cysteine pairs form inter and intra chain disulfides. Like if we talk in helix, single helix or double helix, inter and intra disulfide bonds can be formed for crucial hormone activity. And when we go to the third case study, hydrophobic co-mutations, which are majorly observed, like long bulky hydrophobic residue. Hydrophobic residue here generally refers to the non-polar which do not form hydrogen bonds, or they lack the strength to form hydrogen bonds, and often defolding are replaced by the polar ones, or which can form strong hydrogen bonds. So the function they need to carry can be carried and does not get disturbed due to the still placed there due to the initially placed there non-polar groups. So coming to the next topic, we have some study strategies and mnemonics for amino acids. This was all the classification we referred to as. First we studied about the polar ones, the non-polar ones, the essential and non-essential, the amino acids divided into acidic and basic nature, and also some special categories like how disulfide bonds are formed. Some of them can absorb UV rays. So some basic strategies and mnemonics for amino acids, like you should group mnemonics. You can have a group discussion to enhance or improve or get a better grasp on this topic. And the second, we have active recall and space repetition. It is a kind of game in which cards basically have three letter abbreviation or one letter abbreviation, and the main structure you need to connect three all to win the game. And the third is the practice drawing structures. You need to draw the structures of these 20 majorly used amino acids in order to have a better grasp on their structure. Like it can be done in free time. Just take a pen and paper and write them down, and the suggested problems predict ionization states at given pH values. This can be a little difficult, but with time it will definitely benefit you. So going on, this was all for today. If I had helped you, thanks a lot. That will be a wisdom. That will be a good. If I had helped you today, that will be a positive review for me. So the concise recap, I have already told you about everything, like the concise recap about the amino group, carboxylic group, how they are bonded together by the side chain to form amino acids, the pKa values and the charges they carry, special residues, and the further reading we have references of certain books which can be used to enhance your knowledge. Thank you everyone. That's for today. Myself Param